All right, hello everyone. Welcome to another video. This is going to be on DNA replication and transcription. My previous video, I mentioned that I would do transcription and translation, but I knew that replication is just as important to cover, and it also gives some insight into transcription. So I figured that we would do replication and transcription first, and I would make a separate video on translation. But let's get into it. All right, so what is the basic idea of DNA replication? So we basically use a template strand from our original DNA in order to create a new complementary strand. And that leads into the term of semi-conservative. Semi-conservative basically associates with the fact that we're using our original DNA strand as a non-coding strand. And I believe it is also called an antisense strand, but I don't quote me on that, I believe, but it should be called that. Anyways, we use a very just the original DNA strand, an old strand, and we construct and we use that for our new strand that is being built. So keep that in mind. Semi-conservative. It's not, it's not totally like we're making a new strand on both sides, but we have an old strand and a new strand. And there are several enzymes in play: DNA helicase, DNA polymerase, topoisomerase, DNA ligase, and primase. We'll go deep into them both, in them all, and we'll see the function of all of them and how they play out in the process. So let's get into that. All right, DNA replication. My camera up there. So what happens first with DNA replication? So we have this original DNA strand, right? It's all coiled and we have all the bases connected by hydrogen bonds. And I call that the rungs of the ladder. So what we need is we need DNA helicase. DNA helicase will come in. It will break the hydrogen bonds between those nitrogen bases. And what this happens as a result of this, if you see on the bottom left hand of my screen where my cursor is, that's what DNA helicase does. It breaks up that coiling. It unwinds the DNA. But we also have another enzyme as DNA helicase is doing its job, and that is called topoisomerase. Basically, topoisomerase will prevent the severe unwinding of the DNA. Basically, it keeps it intact because we don't want our DNA to all of a sudden unwind, and we don't want we don't want two separate strands, of course. So DNA helicase, again, will just break the hydrogen bonds between the bases, and topoisomerase will just prevent the severe unwinding of the DNA. And if you see here on the bottom right hand, or just the bottom of the text that I have, you see there's a directionality of the DNA, 5 prime, 3 prime, and segments of DNA, and it's called Okazaki. We'll dive deep into that in the next slide right now. Okay, my camera down. All right, so there's a lot going on here, yes. But let's examine the bottom left-hand photo there. That shows like the leading strand and lagging strand, right? So, and we also have two main enzymes in play: DNA polymerase and primase. Primase is not labeled here, but what it does is that it creates what is called a primer. Okay, so a primer, what is that? The primer that primase creates is basically a starting point. It's an RNA. What DNA polymerase does is it uses that as a starting point to construct the new strand. And although you're asking, well, primase, well, the primer is RNA because the primase was an RNA enzyme. Yes, but when we complete the replication process, DNA polymerase will come back and replace that primer with a DNA code. It replaces, instead of it being RNA, it will become DNA. So we have everything intact and complete. But keep in mind that the primer is a starting point for DNA polymerase to construct the new strand. And I also wanted to make note of the directionality. Now you see here, there's a leading strand and there's a lagging strand. So let's look at the leading strand. So the green shows the new strand being built because it has that arrow. And it's being built in the 5 prime to 3 prime direction, right? But keep in mind, it's being read in the 3 prime to 5 prime, but it's being built in the 5 prime to 3 prime direction. Very important. the directionality is very important to make sure you know which one is which and distinguish between the two. Now let's examine the lagging strand. It's the total opposite of the leading strand in terms of directionality. But also you can see it's segmented. That's weird. Basically, uh, and also keep in note that the leading strand was moving towards the fork, that replication fork, because it's like it's shaped in the fork. But the lagging strand is being built away from the fork and being built in segments. Now, these segments, we don't call them segments. There's a name for the separate segments of DNA, and those are called Okazaki fragments. But in the end, you're probably asking, how do these segments in the end create the complementary strand? Because they're not complete. But that's where DNA ligase comes in. Now let's go to the top right-hand corner of my screen. Hopefully you can see my cursor where I'm circling DNA ligase. That enzyme will connect the segmented DNA strands together, which are part of the lagging strand. And after that, we have our completed replicated DNA. So keep in mind that DNA ligase will connect those Okazaki fragments that pertain to the lagging strand, and the directionality of the leading strand is 5' to 3', and the leading strand moves towards the replication fork. Those are the very main important things you need to know and make sure that you have down. Okay, so after we covered replication, let's look at the basics of transcription. So it is similar to the replication process, but we will have a particular enzyme mainly in play, and that's RNA polymerase. What RNA polymerase does is that it elongates the DNA to create an mRNA strand. So it essentially carries a code for a particular protein, right? In the end, that's translation. But basically, we need the RNA polymerase enzyme to create that mRNA. And we will examine that process further in detail. Okay, so let's look at this image here. But let me first mention that RNA polymerase will need what are called transcription factors. What these transcription factors are is basically they are helper proteins. RNA polymerase needs these transcription factors in order to bind to the DNA. It cannot solely by itself just bind and create an mRNA strand. We need these proteins first in order for RNA polymerase to bind to the DNA. And also, this is also very important, there is a specific location on the DNA where RNA polymerase will bind and where all the transcription factors will also be there and bind too. And that is called the promoter. That is called the promoter. The promoter is known as the Tata box, thymine, adenine code. But basically, again, that's the code. That's the location where the transcription factors will bind and be located, as well as RNA polymerase coming in to elongate the DNA and create the mRNA strand. And also, there's a similarity to the replication process here. The mRNA strand is basically in the same directionality as the leading strand. So it is being built in the 5' to 3' direction, but being read in the 3' to 5' direction. So mRNA has the same directionality as the leading strand. And you can see here that in the picture that shows the process, you can see the transcription factors, those big proteins. And the promoter region is that orange piece of the DNA. And then RNA polymerase is also that big enzyme there. It's huge. And it forms this bubble. And that's where elongation comes in. Elongation is that replication bubble that RNA polymerase creates when it's creating that mRNA strand. And keep in mind that we use this non-coding strand, the template strand, for the mRNA as well, which is similar to the DNA replication process. Okay, so let's do a comprehensive review. So first thing is that DNA replication is semi-conservative. So again, consider this as we have an old strand from our original DNA, and we have a new strand being built. Leading a strand reads through the 3' to 5' direction, but it is being built in the 5' to 3'. So it's being built in the 5' to 3', but being read in the 3' to 5'. An RNA primase will add a primer that will eventually get replaced by DNA, or even though it is RNA at first, so that DNA polymerase can build off of it as a starting point. For transcription, we will need transcription factors, forgive me for that spelling, which will allow RNA polymerase to bind and code for the mRNA. Those transcription factors, without those transcription factors, we can't have transcription. There is no transcription that is possible. And keep in mind that also that we have a specific location where the transcription factors bind to, which is a Tata box. And that is how transcription will be conducted and facilitated. So with that being said, thank you guys so much for watching this video. I hope you guys enjoyed it. I hope it was better than the last one, actually. And if you guys have any further questions, feel free to contact me. And in the comments, please, I value your feedback. So let me know if I did well this time, I did not do so well. I really value your feedback again. And keep in mind that I will be making videos not only on biology concepts, but as well as calculus, physics, English, environmental science, as well as some SAT tips that I have for myself. And I want to communicate that to you guys. So thank you so much, guys, for watching again. And I will see you in the next one.